Okay, students, I'm just going to run through this activity that you did and make sure you understand what we did here. So this part, looking at the spinal muscular atrophy, was a way to teach you some of the biotechnology techniques that we have that you didn't already learn about in lab. So just expanding a bit upon that. So the first technique is called quantitative PCR. So as it describes here, we're basically trying to figure out what messenger RNAs and in what quantity they're being expressed in the cell. So we want to know specifically in this one if the SMN1 mRNA is being expressed, and if so, how much of it in these patients. And actually, we're looking for SMN1 and SMN2. So basically, you isolate the mRNA from a cell, and then you use a reverse transcriptase to make it back into DNA so you can use it in PCR. And then when you do the PCR, you have all the little nucleotides, the A's, C's, T's, and G's. They have a little fluorescent dye on them, so they start to show up fluorescently in the material so that you can tell when you're accumulating things. Okay, and so they did this on a bunch of patients, and let's see if I can zoom in here. We'll get even bigger here. So they did it on a bunch of patients, so you can see that A is a normal homozygous person, so they've got good SMN1. B is a heterozygous person, so half SMN1 and half not, the SMN2. And then C is 2 affected patients who have only the SMN2. And so what you see is this is time. So over the cycles, we're not seeing much fluorescence. And then all of a sudden we start to see fluorescence. Boom! And it goes up. And same thing with B and C. D is a control. And so what you see is that the sooner it starts to take off, it means there was a lot of mRNA to copy to begin with. And so from this, you can see that C, these patients, they have less of the mRNA, whether it's SMN1 or SMN2, because we use primers that'll amplify either one. We see that they just have less of it. So the cell is expressing less of the messenger RNA than it should be. And then the other thing you can tell from this activity, or from the qPCR, is it also tells you the melting temperature. So remember, PCR, the very first thing is to denature the DNA, to heat it up so it'll melt so we can copy each side. And this is showing you the melting temperature for each of these patients. And so the sooner it melts, the shorter the piece of mRNA or whatever you're amplifying is. And so you can see in our mutants, that's what the M stands for. In our mutants, they melt at a shorter temperature than our wild type people. And then if you're a heterozygous person, you get 2 bumps for the melts. And so from this, we can see that the DNA piece that is in an affected person is smaller, is shorter than the DNA piece in an unaffected person. And so we somehow know it's shorter. So we're trying to, again, remember, we're trying to figure out why a silent mutation would cause any kind of error at all, because it's silent, right? It should code for the same amino acid. So the next one was the Western blot, and the Western blot is looking for proteins. So we run them on a gel, and the proteins get stuck at a certain place on the gel. And then we have an antibody that matches that protein, and then an antibody that matches that glows. So we can see where it is and how much of it is being expressed. And so if you look at the people, this is a control, and you look at normal people, how much SMN protein they make a ton. Whereas in an SMA patient, you see that it's there, but it's faint. So they make very little protein. So we already know they make this mRNA that's somehow shorter, and then somehow that translates into less protein altogether. The IP90 is just a control. Okay, so we know, number 1, that they produce less of the mRNA, number 2, that the mRNA is somehow shorter, and number 3, that somehow leads to less protein being produced. Okay, so then we go to the the northern blots. And yes, it was named after one of them. I can't remember. It's either Dr. Western or Dr. Northern. And then every other blot after that is just making fun of them. And there's a southern blot, and there's a southeastern blot, and a northwestern blot, and all of these others. Anyway, so the northern blot is looking for mRNA. So instead of protein, it's the same idea. But instead of protein, we're looking for mRNAs. So we isolate all the mRNAs from the cell. We run it on a gel, so it will run by size. And then we use, instead of an antibody that would detect a protein, we use a little piece of RNA that is the opposite sequence, so it'll complementary bind to whatever mRNA we're looking for, and we have the little labels on them so we can see it. And so again, here we have wild type, meaning a normal person. They make SMN1 and a little bit of SMN2. This is our control. And then our SMA patients, they make no SMN1, which we knew, right? They've got a mutation in that gene, but they make a ton of the SMN2. The other thing you can see from this is size, right? So if it ran faster on the gel, it was a shorter mRNA. So the SMN2 mRNA is shorter. So whatever that mutation is, that silent mutation, it's somehow making the mRNA shorter, which we knew from the quantitative PCR. Okay, so this just confirms it. And then the next piece of evidence is molecular mass. So you could see that we're missing some amino acids on the finished protein. Okay, so somehow, and that makes sense. If the mRNA is shorter, the protein's going to be shorter too. And so the last bit of information is looking at the mutation. So the mutation is right here. This T should be a C, but it's a T in the SMN2 sequence. And if you go down and look at the table, the particular thing that is messed up is this sequence, CAGACAA. So if you go up in the original one, it should be right here. See how it's CAGACAA? But in this one, it's TAGACAA. And so what happens is it fails to see this little piece as an exon, and it cuts it out. We actually end up translating a little bit of the following exon, which is why it's not an exact 13 amino acid missing, but we're missing exon 7. And so that's what makes the protein shorter. That's what makes it not function. And interestingly enough, SMA or SMN is part of the spliceosome. It's part of the thing that cuts introns and exons. So it's almost like what came first, the chicken or the egg. The mutation makes a bad spliceosome, which then in turn badly splices the proteins. And so you end up with the disease. But if you have a ton of SMN2, so some people just have extra mutations, extra duplications of this gene. So you might have 8 copies of it or 4 copies of it, and someone else might only have 2 copies of it. And so if you don't have, if you only have 1 or 2 copies, you're likely going to die very early. It's when you have 4 to 8 copies that you could live into adulthood. But that's not true anymore because we now have a cure, like just recently. Just in the last few years, this has made it through clinical trials. So this came out in 2016. Since then, it's made it through clinical trials. There's several different names of it. We'll talk about them briefly in class. But it's saving these babies, which is amazing. Okay, so that's the overview of that particular activity.